Look at these two organisms. The first reproduces asexually, splitting instantly into two. The second relies on sex, requiring a slow, resource-heavy courtship. Asexual cloning is fast. Every individual can bear young, doubling the population with each generation. Sexual reproduction cuts that reproductive capacity in half, while burning enormous amounts of time and energy just to locate a mate. By every basic law of thermodynamic and biological efficiency, asexual cloning should dominate the planet. Yet almost all complex life relies on sex. To understand why, we have to define sexual reproduction objectively. It is a composite biological technology consisting of two physical steps: meiosis, which cuts a genome in half, and syngamy, which fuses two halved genomes into a new, complete cell. Maintaining the physical infrastructure for those two steps is incredibly expensive. Organisms do not pay that energetic price for the sake of random cosmetic variety among their offspring. Sexual reproduction is an algorithmic defense mechanism. To understand why it mathematically dominates cloning, we must reverse engineer this biological machine, from the scale of global populations down to a few nanometers. The blueprint traces back 2 billion years to the last eukaryotic common ancestor, or LECA. It was a symbiotic merger. An archaeal cell swallowed an aerobic bacterium, harnessing it to utilize oxygen. That bacterium became the mitochondrion. This energy source had a lethal byproduct. Oxidative metabolism inside these early mitochondria released highly volatile reactive oxygen species, which flooded the cell, striking and severing the organism's DNA. These double-strand breaks were catastrophic. If a single strand of DNA is damaged, the cell can read the opposite strand to fix it. But when both strands are severed, the genetic sequence in that location is entirely lost. To survive their internal energy production, early eukaryotes required informational redundancy. They solved this using homologous recombination. The cell borrows an intact complementary chromosome, aligning it with the severed strand to serve as a physical template to rebuild the gap. Eukaryotes required this oxidative damage repair mechanism to survive their own metabolism, making early sex a mandatory requirement for cellular survival. This ancient repair mechanism eventually scaled into a population-level defense system called the Red Queen hypothesis. We observe this dynamic in New Zealand's lakes, home to the Potamopyrgus snail, a species capable of reproducing both asexually and sexually. The shallow lake edges are dense feeding grounds for ducks. Through their droppings, ducks transmit a sterilizing trematode parasite directly into the snail population. In the deep waters where ducks rarely feed, the infection rate drops. Down there, asexual snail clones dominate the ecosystem. This chart compares reproductive strategies by water depth. In the deep water, cloning works. But in the highly infectious shallow zones, the clones crash, and the sexually reproducing population takes over. We see the same survival advantage in Atlantic salmon. Comparing wild salmon that choose their mates to commercially bred hatchery salmon denied that choice, the hatchery offspring carried significantly higher parasitic roundworm loads. Parasites possess short lifespans and rapid mutation rates, allowing them to exploit static genetic sequences. Host species use sexual reproduction to constantly shuffle their genetics, ensuring their offspring remain a moving target against rapidly evolving pathogens. Rapidly adapting parasites are an immediate threat, but asexual populations face a second, slower threat: long-term genetic decay. Every time a genome replicates, random copy errors occur. Because these errors happen across billions of base pairs, the odds of a second mutation perfectly undoing the first one are statistically zero. Without a way to swap out damaged code, an asexual lineage only accumulates deleterious mutations. This is Muller's ratchet. Once a mistake is locked in, the gear cannot turn back. We can measure this decay in Daphnia, microscopic water fleas that maintain both sexual and clonal lineages. When sequenced, clonal Daphnia carry a significantly higher proportion of amino acid-altering mutations. Over time, their genetic burden increases. Meiosis physically breaks apart and recombines chromosomes, isolating and clearing harmful mutations. It's the only mechanism capable of turning the evolutionary ratchet backward. To see exactly how the cell executes that shuffle, we must leave the population scale and dive directly into the nucleus. Genetic recombination is handled by two specific Rec A homolog proteins. RAD51 and DMC1. Their job is to bind to the ends of a broken DNA strand, search for a matching sequence, and catalyze the exchange. For years, our understanding of these proteins was limited by blurry, wide-field light microscopy. Due to low resolution, textbooks adopted the asymmetric loading model. It assumed RAD51 gathered entirely on one side of a break and DMC1 on the other. This impossibly required the cell to perfectly sort and load specific proteins to one side while ignoring the other. Advanced imaging reveals a symmetric architecture that standard microscopes simply could not resolve. The breakthrough came via DStorm, direct stochastic optical reconstruction microscopy. By bypassing the diffraction limit of standard light, DStorm allows researchers to track individual fluorescent molecules, mapping the physical layout of these proteins. When scientists looked at RAD51 and DMC1 through DStorm, they found the proteins don't segregate. They actually appear in clustered, overlapping pairs. The two ends of a single DNA break are physically separated by a gap of up to 400 nanometers. That gap is highly regulated. Even if researchers block chromosome synapsis or halt strand exchange, that distance holds firm. Furthermore, what looked like a single large clump of protein is actually a highly fragmented cluster of multiple distinct substructures. RAD51 and DMC1 do not pick sides. They symmetrically co-occupy both ends of the meiotic break. Under super-resolution, these clustered protein substructures measure just 115 nanometers across. Once you subtract a bulk of the chemical antibody markers used for imaging, the true core protein structure is only 55 nanometers long. In genetic terms, a 55 nanometer filament covers roughly 100 nucleotides, exactly 5 turns of the DNA helix. When a double-strand break occurs, the cell resects the DNA, exposing a massive single-stranded tract of about 800 nucleotides. Instead of wrapping that entire tract in one continuous filament, the cell uses multiple ultra-short filaments scattered sparsely across both ends of the break. This sparse, fragmented assembly defines the true recombinosome, a symmetric machine that executes genetic shuffling with high fidelity across both ends of the break. To understand why sex persists, you have to draw a straight line from that 100 nucleotide filament up to the macroscopic environment. The long-term persistence of eukaryotes depends on the subdiffraction physical precision of this system. This fragmented, symmetrical co-occupation is what physically enables high-fidelity homologous recombination. This molecular engine allows host organisms to shuffle their genomes, keeping them ahead of parasites in the Red Queen's race. It provides the mechanical action required to excise damaged genetic code and successfully reverse Muller's ratchet across generations. Sexual reproduction operates at a metabolic deficit, but it pays for that cost with long-term viability. What began as a repair tool for a single cell became the physical requirement for complex life to persist across geologic time. Observe this biological environment. Clonal organisms simply divide, multiplying their population rapidly. Sexual organisms, meanwhile, must halt replication to engage in a slow physical union. Clonal organisms transfer 100% of their genome, progressing to 4 exact copies. Sexual inheritance forces parents to discard half their genetic material, yielding a strict twofold cost. This baseline penalty is compounded across generations by the energy required to find a mate. This presents an evolutionary paradox. A reproductive strategy operating with a continuous 50% handicap should be rapidly outcompeted and driven to extinction. Yet sexual reproduction dominates eukaryotic life. Sustaining a permanent twofold fitness penalty requires an explanation. It demands either an absolute, overwhelming survival advantage that makes up the deficit, or population dynamics that stabilize the handicap through nonlinear mechanics. The prevailing consensus is the Red Queen hypothesis. Like the Lewis Carroll character, organisms must constantly run just to stay in the exact same place. They must continuously shuffle their genetics to survive against rapidly mutating parasites. Field data supports this mechanic in isolated scenarios. Biologist Robert Fryenhoek spent decades observing Mexican poeciliid fish. Some populations reproduce sexually, others asexually, and both are continuously targeted by a parasite that causes black spot disease. This graph plots the sexual fish against the parasite. Through continuous genetic shuffling, the sexual replication curve dynamically outpaces the fluctuating parasite mutations. Meanwhile, the static asexual genome offers a fixed target. Once the parasite cracks their defense, the clonal lineage collapses. When we apply mathematical rigor to this scenario, the model hits a limit. To offset a 50% penalty, the Red Queen requires an extremely high mutation rate, at least one deleterious mutation per generation. Yet when we measure real-world species, the actual mutation rates are frequently too low. The math explains specific crises, but it fails to account for the universal maintenance of sex. Data across various species suggests that genetic variation appears as a secondary byproduct of a much older structural necessity. To find the actual origin of sex, we have to look at the primordial RNA world. Early genetic material existed as fragile, single-stranded RNA molecules in a volatile environment. These genomes were constantly bombarded by reactive chemicals and fragmented by radiation. This diagram shows the very first mechanistic iteration of sex. To survive lethal damage, early replication machinery developed a copy choice protocol. When the enzyme hits a damaged lesion on one strand, it executes a physical switch to a parallel, homologous strand to continue transcription. We see this mechanism operating today in simple DNA viruses. When a bacteriophage like T4 is exposed to ultraviolet light, the radiation shears its genome, creating lethal double-strand breaks. If a single damaged virus infects a host cell alone, the replication machinery encounters the breaks and fails. The virus produces no viable progeny. This illustrates multiplicity reactivation. If two lethally damaged viruses co-infect a single host cell, their fragmented genomes physically interact. They align their homologous sequences and exchange undamaged segments. The fragments seamlessly merge, reassembling into a single functional genome, highlighted as the output. Biologists define this viral recombination as sporadic sex. This is a highly specific protocol triggered by genomic stress, where error correction takes precedence over the generation of genetic diversity. At the foundational molecular level, homologous interaction operates as an error correction mechanism. The pressure to repair lethal damage appears as a much earlier selective force than the pressure for variation. This molecular repair protocol scales up to complex eukaryotes. The life cycle of the single-celled protozoan Paramecium tetrarelia demonstrates what happens when an organism relies solely on asexual division for too long. As the paramecium undergoes successive asexual fissions, it experiences clonal aging. Lethal DNA damage inescapably accumulates in its active vegetative macronucleus. If the organism divides asexually 200 times, the accumulated damage reaches a fatal threshold and the lineage expires. This animation shows the meiotic rescue protocol triggered by cellular starvation or age. The organism dissolves its highly damaged macronucleus. It relies on its protected micronucleus, which undergoes homologous recombination and generates a pristine, newly formed macronucleus. The architectural core of eukaryotic meiosis evolved to locate and repair double-strand DNA breaks. Specific pathways like synthesis-dependent strand annealing perform this error correction without the absolute requirement of crossing over or generating novel variation. Molecular repair explains the physical mechanism of sex, but the population mathematics must still explain how this repair strategy survives a 50% reproductive penalty in the wild. This game-theoretic model maps the evolutionary stability of sexual populations, identifying a sharp boundary exactly at a reproductive cost of 0.5. Stability requires assortative interactions. Populations achieve equilibrium when organisms evolve sensory tools to detect the sexual state of others. These sensory feedback loops cause individuals to dynamically adjust their reproductive mode based on their environment. The interactions cease to be random. This non-random assortment effectively traps the population in an evolutionarily stable strategy. The math confirms that sexual reproduction does not require a net fitness gain to survive. Assortative behaviors mathematically lock in the repair strategy, maintaining the mechanism even when the energetic cost is strictly twice as high. This ancient recombinational repair protocol is continuous through the highest orders of biology. We see it operating within the modern mammalian germline. This diagram shows a phenomenon known as prophase 1 arrest. Mammalian oocytes pause their development, locked specifically in a 4C meiotic configuration. This state distinctly highlights the availability of 4 separate parallel homologous sequences. This 4C configuration provides continuous, redundant templates. As the organism ages and metabolism produces oxidative free radicals, those templates are used by cellular machinery to repair double-strand breaks in the DNA. This extreme developmental arrest exists for a single architectural purpose. It provides the physical framework necessary to protect the integrity of the female germline against terminal degradation before reproduction occurs. This flowchart diagrams 2 competing explanations for the existence of sex. The primary central trunk of the system is the response to DNA damage, which leads to recombinational repair, resulting in a viable germline. In this framework, the Red Queen hypothesis functions as a secondary branch, a beneficial downstream consequence of the primary repair system. Genetic variation provides a defense against pathogens, but it grows out of the primary trunk of recombinational repair. The survival of complex lineages is rooted in the mechanical precision of the repair process, which restores genomic integrity at the molecular level. The 50% loss of genetic transfer represents the immediate toll required to operate a multi-parental error correction system. This process ensures the survival of complex life by preventing the catastrophic degradation of the genome.